Rotem Sorek is one of the most famous, prominent, and highly accomplished microbiologists in the world, well recognized for his work on the interactions between bacteria and the viruses that infect them. His discoveries on the immune system of bacteria have opened completely new fields in microbiology and have made paradigm shift in virology. Professor Sorek's scientific excellence is demonstrated both by his extraordinary creativity and innovation and by his high level of productivity. Now it comes actually very relevant to the previous lecture. Apart from his impressive achievements in basic science, basic research, Rotem Sorek is also a very prolific and innovative inventor with 40 patents and patent applications. 9 of these patents have been licensed to biotech companies. Technologies invented in the Sorek lab were the basis for the establishment of 3 startup companies already. What can be more appropriate as a bridge between the previous keynote presentation and the Rotem Sorek? Rotem, I would like to invite you for your lecture, and I would like to invite you all to congratulate again to Rotem for this prestigious prize.
Thank you. Thank you very much for this enormously generous introduction. I'm very honored and humbled to receive this very highly prestigious prize from the HFSPO. And I apologize for not being able to participate in the meeting in person. The original plan was that I will come and join you in person, but the situation in my country these days makes travel more difficult. So I would hope to entertain you with some science in the next hour or so. Let's go directly to the talk. Indeed, what we do in the lab is we're studying the immune system of bacteria. And the immune system of bacteria, you know, some people know it as the CRISPR system, but we studied the immune system of bacteria beyond the CRISPR system. And so I guess that you'd ask yourself, why would bacteria need an immune system? We're thinking about bacteria usually as something that our immune system fights against very frequently. But in fact, bacteria themselves are facing infections by viruses all the time. And the viruses that infect bacteria are called bacteriophages or phages. And these viruses, like any virus, they recognize something on the surface of the bacteria in order to identify their host. And then they inject their DNA into the bacteria. And in the bacteria, this DNA forms, you know, it's like a computer virus. It cheats the bacteria and takes all the resources from the bacterial cell to replicate itself, and then form new virions within the bacteria. But then, after a while, they kill the bacteria and emerge out of the bacteria to search for new bacteria to infect. And so these viruses, most people don't think of them, but in fact, they are by far the most abundant viruses on Earth. There are more viruses that infect bacteria, more phages in the world than bacteria in the world. So about 10 times more phages than bacteria in the world. It was estimated that if you take all phages in the world and weigh them, put them on a weight, they weigh 4 times as much as all humanity itself. So they have big role in the biology, the life cycle on Earth. Now, since there are so many phages, one may be surprised that bacteria even survive, because phages infect very efficiently. And to defend themselves against phages, bacteria have evolved an immune system. And the immune system that most of us knew about, I guess, until about 5 years ago, is comprised of 2 main arms. One is the CRISPR-Cas system, which I'm sure that many people in the room have heard of. It's an amazing immune system that allows bacteria to memorize past infection of phages by taking pieces of the phage genome and storing these pieces into the bacterial genome. And then the system uses these pieces to block infection by future phages by cleaving their DNA. That's an adaptive immunity arm, very sophisticated and fascinating defense system that bacteria have. Another arm of immunity the bacteria have, the innate immunity arm, comprises restriction modification systems. And these are systems that were discovered very early on in the 70s, maybe even earlier. And these comprise an enzyme that cleaves the incoming DNA of the phage, the restriction enzyme, and it cleaves in a specific motif. And the restriction enzyme in the bacterial cell will always be accompanied by another modification enzyme that modifies the same motifs on the bacterial genome, usually by methylation, so that the restriction enzyme will not cleave these motifs to prevent autoimmunity. So these were the main immunity arms that were known to us until about 5 years ago. But at some point, I mean, we studied CRISPR for many years, and at some point we started ask ourselves whether there might be new defense mechanisms that we are not aware of yet. And the reason we thought that there might be new defense mechanisms is that the CRISPR-Cas system was discovered really late in the history of microbiology. Think about that. Phages were discovered in 1915, more than 100 years ago. And for a while, phages were basically the only entity that was studied to decipher how molecular biology works. The fact that DNA is the genetic material, transcription, translation, they were all studied by phages infecting bacteria. And still, CRISPR was discovered really late, only in 2005. And the reason that CRISPR was discovered so late is that it's not very active in E. coli. As you know, E. coli is the main workhorse as a model organism, model bacteria that people studied bacteria and phages. So we started thinking maybe other bacteria that are not model organisms have other immune systems that are not CRISPR, and maybe we can discover them somehow. Now, why even be interested in new immune mechanisms? Why do we care about them? So the reason, first of all, is that if we want to understand the arms race between bacteria and phages, we need to know, from the evolutionary perspective, we need to know how bacteria defend themselves and how phages can overcome this defense. But from a more practical point of view, if you think about previous discoveries of immune systems, you see that the discovery of a novel immune system and understanding how it works in the past led to revolutions in biology. So the discovery of restriction enzymes led to the ability to genetically engineer DNA. The discovery of CRISPR now allows us to engineer genomes. And if you go beyond the immune system of bacteria, then you see that the discovery of RNAi in plants and in animals led to a revolution in the ability to silence genes in humans. And the discovery of antibodies allows us to do many things with these antibodies, like pulling down stuff, doing diagnostics, etc. And the reason why immune systems are so very frequently beneficial, becoming beneficial tools, is that an immune system, by definition, needs to identify some molecular moiety very specifically in order to differentiate between self and foreign, and then do something to that molecular moiety, like cleaving it or removing it or pulling it down. And so we think that if we discover new immune systems, they might in the future become new molecular tools that will do yet additional revolutions in biomedicine. But the question is, how can you identify defense systems in a systematic way, given that all these defense systems have been discovered in the past just by chance? People studied one thing and they bumped into one of these defense systems and discovered it further. So we try to develop systematic methods to find new defense systems. And we had these ideas since 2010, and we tried several methods that didn't work, so they failed. I will not present them to you. But what seemed to work very well eventually stems from a very weird behavior of bacterial defense systems when you look at bacterial genomes. So if you look at bacterial genomes, you see that defense systems cluster next to each other in so-called defense islands. So what I'm showing you here, for example, is a piece of this genome. It's a genome of a bacterium called Alisiclibacillus. And you see a small piece of the genome. Each box here is a gene. And you see that genes that encode CRISPR-Cas are encoded very near genes that encode restriction enzymes. Here is the restriction enzyme. This is the modification enzyme. This is the specificity subunit. And that's something that we see again and again in many different bacterial genomes. Defense systems that are marked here by this orange color are present next to each other very frequently in these defense islands. So we started thinking whether if we look for genes of unknown function, cassettes of genes that are not known yet to participate in defense against phage, but they are present frequently next to CRISPR or restriction enzymes. When you look at many genomes, we might find new defense systems. And so Shani Doron, when she was a PhD student in my lab, she analyzed tens of thousands of microbial genomes to look specifically for cassettes of genes that are present next to CRISPR and restriction enzymes in a statistically significant, enriched way. And so, surprisingly to us, we discovered many, many predicted systems that we did not expect. We thought we might discover 1 or 2 or 3 new defense systems, but in fact, we had many, many predictions. And so, of course, these predictions remain on your computer until you devise a way to test them to see whether they are indeed correct. So we, in parallel, devised a validation strategy where if we have a candidate new immune system, we look at all the donor bacteria in the microbial databases and select bacteria that encode the system that are as phylogenetically similar to the ones that we work with in the lab, Bacillus subtilis and E. coli. These are bacteria that normally do not have many functional defense systems. And that's the reason why they became a model organism. You can insert DNA to them without any problem. Then once we select the donor organism, we take the defense system and we synthesize the DNA, and then we install this DNA within the genome, either of Bacillus or of E. coli. And now we take these engineered bacteria and infect them by an array of phages, different kind of phages, like RNA phages, DNA phages, double-stranded DNA, single-stranded DNA, etc. And now we can see whether the defense system or the candidate defense system provides defense against one or more of these phages. And I think by now we tested more than 500 different defense systems or candidate systems this way and cloned more than 1 million bases of DNA inside model organisms and tested them this way. So it was a huge effort of many different people in the lab. And again, very surprisingly to us, we found not 1, not 2, but many, many, many defense systems that bacteria encoded and that were not known to us until we started these studies. So what you see here in the picture is a figure from one of our first papers in 2018. Let me try to walk you through what you're seeing here. So this is a defense system we found. It contains 2 genes. One has a domain of unknown function, and the second has a domain of helicase, meaning that it somehow deals with DNA or RNA, binds DNA or RNA or cleaves it, we don't know yet. And we took it from Bacillus cereus, a close relative of Bacillus subtilis. We cloned it into Bacillus subtilis, and we infected Bacillus subtilis by 10 phages from different families. And whenever you see a marked color here, it means, the red color means that the system defended against the specific phage. So here you see that we had to infect by about 1 million times more phages in order to start seeing infection if we clone the defense system into the bacterium. So this is a pretty strong defense. And so since 2018, focus in my lab shifted from bioinformatic analysis of genomes into biochemistry and trying to decipher the mechanisms of action of these immune systems that we discovered. And one of the things we noticed pretty early on is that some of these defense systems encode domains that sounded familiar to us. For example, TIR domain is known to participate in immunity against viruses and bacteria, but not in the immune system of bacteria. The TIR domain is an acronym of Toll Interleukin Receptor, and that's a domain that participates in our own immune system, the immune system of humans. And so we started seeing a connection between the immune system of bacteria and the immune system of humans. So there seems to be an ancient evolutionary origin for innate immunity that is shared from bacteria to humans. And then we started asking ourselves whether there are other components of our own immune system, our own innate immune system, that evolved from the bacterial antiphage defense system. And the answer was yes. I'm going to tell you a bit more about this aspect of our work. So here's one mechanism that we have in our own cells. So many cells in our body, pretty much any cell in our body, has the ability to sense that it has been infected by viruses and do something about this virus infection, even if this cell is not an immune cell. It's called the cell autonomous innate immune system. And one of the key pathways of immunity is a pathway that's called the CGAS-STING pathway. It comprises two proteins. One is called CGAS, and that's a sensor for double-stranded DNA that is present in the cytoplasm. So as you know, DNA in the cytoplasm in human cells is illegal because you have a nucleus and DNA has to be in the nucleus. So if a DNA is sensed outside of the nucleus in the cytoplasm, it means, or it is interpreted as if the cell has been infected. And so once this protein senses DNA in the cytoplasm, it produces a unique signaling molecule. And this signaling molecule is called cyclic GMP-AMP or CGAM. It comprises two nucleotides that are bound head to tail to each other in a cyclical form. And this molecule accumulates in the cell and binds a second protein. It's called STING, the acronym of stimulator of interferon genes. And the acronym comes because once it senses the signaling molecule, it signals to the nucleus to activate the expression of interferon and of other defense genes that the cell uses in order to defend against a virus. Now, the same molecule was known to be produced by a similar protein, a similar CGAS protein, also in bacteria. And that was known since 2012. It was a publication by the John McAlanus lab at Harvard Medical School. But the function of this molecule and of this enzyme in bacteria was completely unknown until recently. And why am I telling you this story? I'm telling you this story because when we look at the homologue of this protein within bacterial genomes, we find it very frequently encoded next to CRISPR in Vibrio cholera, for example, or next to restriction enzymes, or next to other defense systems that we identify using our pipeline for detection of defense systems. So we have this CGAS protein. CGAS is the acronym of cyclic GMP-AMP synthase. That's why it's called this way. You see that it's present not only next to these defense systems in different microbial genomes, it's also found within an operon of 4 genes. And this operon, one of the genes in the operon encodes for a phospholipase, something that cleaves membranous moieties. And so because of this enrichment of this operon next to defense systems in microbial genomes, we hypothesized that it might be an antiviral defense systems in bacteria. And so as I told you, once we suspect that an operon functions as an antiviral defense operon, we take it. In this case, we took it from Vibrio cholera, from other bacteria that encode it, and then we cloned it into bacteria, into E. coli of the lab that doesn't have this operon usually, or it's not encoding this operon. And then we infected it by an array of phages, and we found pretty strong defense. So that's a very typical experiment we do in the lab. It's called the plaque assay. But you see here, the lighter color are bacteria that grow on a Petri dish. And then we infect these bacteria by increasing amounts of phages. So we put a few phages here, about 10 phages here. And when the phage touches one bacterium, it replicates within that bacterium. And then after about an hour, it breaks open the bacterium and 100 phages emerge from that bacterium and infect the nearby bacteria. And this process goes on. And when we come in the morning, we see areas on the Petri dish that are cleared from bacteria. These are the darker areas here. This means that the phage infected that bacterium. So this is a plaque. And we put here, 10 plaques, 100 plaques, 1,000 plaques, 10,000 plaques, etc. And what you can see is that when we clone the defense system within the bacteria, then we need to infect the bacterium with about 10,000 more phages in order to start seeing some infection, meaning that the system provides pretty strong defense. This work was done in my lab by Daniel Cohen and Sarah Melamed. Daniel Cohen was a master's student, and Sarah is a gifted senior technician in the lab. So this discovery took us into a biochemical adventure, I should say, that led us to understand how this system works to protect bacteria against phages. And here's what we discovered. So what we found is that the bacterial CGAS, the homologue of the human CGAS, is expressed in the cell at all times, but is not active. It becomes active when a phage infects. It senses something that the phage brings, This is not the DNA of the phage because cytoplasmic DNA is present in all bacterial cells. There's no nucleus there. Something else that the phage brings, maybe an RNA of the phage, activates the bacterial defense protein, the SIGAs, which produces the signaling molecule. And once the signaling molecule is produced, it binds the second protein that I showed you in the same operon, the phospholipase, and activates the phospholipase. And now this phospholipase eats the inner membrane of the bacterial cell and kills the bacteria before the phages have managed to replicate within the bacteria. Now, I know that this sounds very weird. Why would a bacterium kill itself in response to infection? This is a single-celled organism. So what is the purpose of this cell suicide that was mentioned by the first speaker of today? And so let me try to explain why this is actually an efficient way to protect against phages. So we should realize that bacteria in nature live in aggregates. They live in colonies. They either live as a colony on a solid culture, or they live within a liquid culture as a colony. And the colony of bacteria are bacteria that are basically having the same genome. They replicated, one cell replicated into two replicated into four, and all of them have the same genome. Now, at some point, one phage drifts into the colony and infects a single cell. After a short while, it replicates within that single cell, and the emerging phages will infect the nearby cells. And these cells will be killed as well. And eventually, the entire bacterial colony will die. There will be no more bacteria with this process. But if the bacteria has the ability to sense that they have been infected and then kill themselves before the phage managed to replicate in the bacterium, only one cell is getting lost, and the rest of the colony survives. So that's a very, very strong evolutionary pressure for bacteria to encode defense systems that allow them to sense that they have been infected and then kill themselves in response to this infection. They're unexpected, but very efficient. And now we know that more than 70% of all bacteria in the world encode at least one system that allows them to sense infection and kill themselves in response to this infection. So during the same time that we did our study with the homologue of the human SIGAs, with the bacterial SIGAs, an amazing study from the Philip Kranz's lab in Harvard Medical School and Dana-Farber Cancer Institute was published. It was the same time. And they showed that bacterial enzymes that look like SIGAs, they're very abundant in bacteria, but some of them produce other molecules, not the same like the human SIGA. So if you look at the phylogenetic tree of this family of enzymes, some of them produce cyclic GMP-AMP, and this is found in many different pathogens that infect humans, but others produce cyclic UMP-AMP, and other homologues produce cyclic UMP-UMP, and other homologues produce cyclic trinucleotides. So a large diversity of these molecules produced by bacteria. So Adi Millman in my lab analyzed the propensity of these enzymes to be present next to known defense systems in microbial genomes. And she found, as I told you, that the genes that encode cyclic GMP-AMP are present very frequently, 62% of the times, next to CRISPR or restriction enzymes when looking at microbial genomes. But very interestingly, all the enzymes from this large family were present very frequently next to known defense systems, and this led us to realize that all these enzymes participate in defense against phages. And these enzymes are cumulatively present in about 15% of all sequenced bacterial and archaeal genomes. So what started by a discovery of a specific system that produces cyclic GMP-AMP to activate a phospholipase is now became a family of defense systems that work similarly, but with variation. In this family, we call this family CBAS, or cyclic oligonucleotide-based antiphage signaling system. And that's from a paper that was published by Adi Millman, a PhD student from my lab that just started her postdoc at the MIT. And these signaling systems comprise two components, basically. One of them senses phage infection and produces a signaling molecule. The signaling molecule could be cyclic AMP GMP, but also cyclic UMP AMP and other signaling molecules. So different bacteria encode different kind of molecules. And this molecule activates the second protein, which is the effector of the system. The protein could be a phospholipase that eats the inner membrane of the bacteria and kills it. It could also be a DNase that is activated by the signaling molecule and cleaves the DNA of both the phage and the bacterium, killing them both in the process. So we call it the SIBA system. So as I told you, the SIBA system comprises different variations of the system, comprise different enzymes, each producing a different kind of signaling molecule. But then we discovered that this is, again, yet only a subset of what really happens in nature, because we discovered other systems that work through different enzymatic capacity, but through the same immune principle. So one system we called Pixar, and this Pixar relies on a different kind of enzyme that cyclicizes C or U. So it's a mononucleotide which is cyclicized. It was discovered by Nitzan Tal from my lab and Ben Morehouse, who used to be a postdoc in Philip Kranzush's lab and now has its own lab in the US. And this is not the only one. There is another defense system that's called Toeris. It was discovered by Gal Ophir and Gil Amitai from my lab. And the Toeris system is relying on yet a third kind of enzyme that produces a weird molecule that relies on cyclic ADP ribose. And so all these systems are basically identifying the phage component, producing a signaling molecule. The signaling molecule activates a second protein, which kills the cell. So there's a huge diversity of such defense systems in bacterial genomes, and we find them in more than 20% of all the bacterial genomes that are sequenced and deposited in the database. In fact, you find these systems in so many bacteria that you start asking yourself, how is it possible that phages survive? How can phages infect these bacteria? Why do we still see phages that infect such bacteria? And so apparently, phages can fight back. And a beautiful study by our dear collaborator, Philip Kranzush, together with his postdoc, Sam Hobbs, showed that some phages produce enzymes that cleave these signaling molecules and do not allow them to reach the effector protein of the bacterium. So they basically interfere in the middle of the immune process, and then the phages can infect. In another study that we did in collaboration with Philip Kranzush, that was led by Erez Yirmiyah, Azita Levitt, and Gil Amitai from my lab, together with Alan Yu from Philip's lab, we found that other phages can produce proteins that very tightly bind the signaling molecules, and again, preventing them from arriving to their destination, to the effector protein that is supposed to kill the cell. So these proteins are produced in massive amounts upon infection by the phage, and they very, very efficiently sequester the signaling molecule and hold it within the protein until the phage finished the replication cycle. So phages can fight back, which is, again, was fascinating for us to see. So let's go back to this so-called eukaryotic cell autonomous innate immune system. The notion of cell autonomous innate immune system is coming to describe the ability of individual cells, and again, not specifically immune cells, but any cell in our body, the ability of these cells to recognize that they have been infected by a virus or a pathogen and do something about this infection. And one of the sensors for viral infection is CGAS, but there are many other sensors like RIG-I or OAS, PKR, many different sensors for viral infection. Once these sensors sense viral infection, they produce some kind of signaling cascade that goes to the nucleus, activates a cascade of expression, and one of the genes that is expressed is called interferon. This interferon is secreted outside of the cell and invites the immune system, like T cells and B cells, to come to the site of infection to do something about the infection. But it also produces a set of genes that are expressed intracellularly that are called interferon-stimulated genes, or ISGs. And these genes are supposed to protect the cell from the infection of the virus. And in many cases, the cell can overcome the infection without needing the support of the immune system like T cells, macrophages, etc. So as I showed you, the SIGA-STING pathway originated in bacteria. So the same pathway has been in bacteria long before it was present in humans. And now I'm going to tell you about a second protein, which is one of these effector proteins. And this protein is called viperin. Viperin is an acronym of virus inhibitory protein. It is produced once cells are exposed to interferon. And it was known for many years that this protein protects human cells from a virus infection. But only 5 years ago, it was discovered why. And the reason, and this is a beautiful mechanism, this was discovered by the Cameron and Almo labs in Mount Sinai, in the US. And they found that the viperin is an enzyme that takes the nucleotide CTP, the normal nucleotide cytidine 3-phosphate, and modifies it a bit. So it removes the 3' OH and puts a double bond instead. So a simple modification of the nucleotide, and it turns the nucleotide CTP into a nucleotide called didihydro-CTP or DDH-CTP. And this molecule is now a poison to the virus replication. And the reason why this is poisonous to viruses is because when the virus replicates, or when anything replicates, basically, it takes the template strand and starts building the new strand. So you take a nucleotide against a C, you take a G, you remove, or here, instead of G, you take against G, you take a C, you remove 2 phosphates from the 3-phosphorylated nucleotide, and you attach the last phosphate to the 3' OH of the previous nucleotide. So now, if the virus polymerase has taken by accident this nucleotide, now you cannot continue to replicate because there is no 3' OH for the next nucleotide to attach. Now, viruses replicate very rapidly, so they make more mistakes. And so the human polymerase can reject this nucleotide, while the virus polymerase occasionally uses it, and then the replication of the virus stops. Now, why am I telling you all this beyond the fact that this is actually a fascinating immune mechanism? I'm telling you this because Ott Bernheim, for my life, discovered that bacteria and archaea contain homologs of the human viperin. These homologs are colored here in red, and this was discovered by Ott when she was a postdoc in my lab. Now she has her own position in Institut Pasteur in Paris. And what she found is that these homologs are very frequently encoded next to CRISPR, restriction enzymes, and other defense systems when you look at different bacterial genomes. So we hypothesized that these enzymes have an antiviral function in bacteria as well. And Ott cloned these enzymes into bacteria and showed that bacteria indeed become protected against infection. But you see here that bacteria, this is E. coli, without the viperin gene, can make huge plaques. This phage T7 infects these E. coli bacteria, makes huge plaques because it replicates very rapidly. After 17 minutes, it finishes replication, so it spreads very rapidly. You see, this is a single plaque. And when the bacteria express the viperin, then you see that the plaques become invisible or smaller. So this red proteins provide protection against phages. So this took us into another adventure, this time of the chemical nature, where we try to decipher how these enzymes work in bacteria. And we found that these enzymes are a family of enzymes that produce a family of molecules. So some of these enzymes produce the same kind of molecule that is produced by the human viperin, the DDH-CTP. But others produce new kinds of molecules that are based on UTP, DDH-UTP, or DDH-GTP. In all cases, the 3' OH is removed from the ribose and a double bond is installed instead. So when you take all the family of these bacterial and archaeal enzymes and you make a phylogenetic tree out of these enzymes, you see that these enzymes are divided into specific clades. Some of the clades tend to produce the DDH-CTP variation. Other clades tend to produce the GTP or the UTP variation. What's very interesting about this tree is that if you take all the viperins from eukaryotes, the human viperin, the mouse viperin, the chicken viperin, even viperin from fungi, and you put them on this phylogenetic tree, you see that they all form a specific clade that emerges from the archaeal clade that produces the DDH-CTP. So this picture tells us that at some point very early on in the evolution of eukaryotes, there has been a single event where most probably an archaeal viperin was acquired by eukaryotes, and then this was the basis of all the evolution of the eukaryotic viperins that we see today. Now, another thing from a more practical point of view that is fascinating to us about this discovery is that it was very surprising to us to learn that bacteria encode antiviral small molecules that can defend against phages. And we knew that bacteria encode antibacterial molecules. Some of the antibiotics that we use are actually produced by bacteria. But now we know that bacteria can produce antiviral molecules. And what's even more interesting is that the antiviral molecules that we take against viruses look very similar conceptually to what we find in bacteria. So these are examples of drugs that we take to protect against viruses that infect us. For example, one of the compounds within the three drug cocktail that people take against HIV is a modified nucleotide, in which case the 3'-OH is modified by, or it's replaced by 3 nitrogens. Here's another drug that people commonly take. This is an anti-herpes drug. This is a drug that people put, on their lips when they have herpes. And it's another nucleotide that's modified so that it doesn't have the bottom part of the ribose. And again, this cheats the virus polymerase. The virus polymerase incorporates this nucleotide within the growing genome, and then replication stops. And here is another very commonly used drug against hepatitis C. And again, it's a modified nucleotide. So we think that now that we understand that bacteria produce modified nucleotides to protect against phages, we are now interested to check whether these modified compounds can actually protect the human body from infection of viruses. There are some preclinical experiments that we're involved with some promising results showing that these molecules, or some of them, can protect our own body against viruses that infect us, or at least our own cells at this point. Let me show you another nice defense mechanisms that we characterize. This was characterized by Nitzan Tal, a very talented PhD student in my lab. Nitzan found that some bacteria can sense that the phage inhibits transcription. So some phages take over bacteria by inhibiting transcription from the bacterial genome. And when the bacteria sense that, they bring into action an enzyme that depletes nucleotides. So it takes the deoxynucleotides that are necessary for the phage to replicate and degrades one of these nucleotides. We found 2 families of enzymes that do this. One family are of deoxy-GTPases that takes the nucleotide deoxy-GTP and separate the 3-phosphate from the nucleotide. So they deplete the deoxy-GTP from the cell. Another family of such enzymes take deoxy-CTP and remove the nitrogen, the NH2, from the base, generating a uracil version. So it eliminates all the deoxy-CTP from the nucleotide pool. Now, when a phage infects a cell, it needs a lot of nucleotides to replicate. It needs the building blocks of DNA to replicate. And when the immune system of bacteria removes one of these building blocks, the DNA polymerase of the phage gets stuck, cannot continue, and the phage replication fails. So for a short while, we were very proud of ourselves for describing a new kind of immune mechanism that was not known before. But then we realized that this was actually discovered before, again, in our own body. So in 2011, it was discovered that a protein in human cell called SAMHD1 restricts HIV infection by degrading the deoxynucleotides. What was found is that when HIV enters, it needs a lot of deoxynucleotides to replicate its genome. But then the SAMHD1 separates the 3-phosphates from the deoxy NTPs and doesn't leave HIV in some cells. So some cells are not infected by HIV because of SAMHD1. And you can see that it's the same kind of mechanism as we see in bacteria. Let me show you another mechanism that is shared between human immunity and bacteria, one of our more recent discoveries. This is Gasdermin-mediated defense. So in human cells, there is an apparatus called the inflammasome that can sense infection by bacteria or viruses. Once it senses infection, it activates a protein called caspase. And this caspase is a protease that activates another protein called Gasdermin by cleaving an inhibitory domain from the Gasdermin. Now the activated Gasdermin goes into the membrane and forms huge pores within the membrane, and then the cell dies. But during its death, it also produces or secretes through these pores interleukins that invite the immune system to help the infected organ. So this is cell death by activation of a caspase that cleaves a piece of the Gasdermin protein, and this Gasdermin forms a pore in the cell. The reason I'm showing you this, you can already guess, is that Anita Vine, a very talented postdoc in my lab that's going to start her position as a PI at the Weizmann Institute soon, she found that bacteria encode Gasdermin-like proteins, so distant homologs of Gasdermin. And in all cases, these homologs are encoded next to a caspase protein or a caspase-like protein or another protein. And also in many cases, this is encoded next to known defense systems. And together with our collaborator, Philip Kranzush, we discovered that the Gasdermin system in bacteria is actually activated by the caspase, and caspase cleaves an inhibitory portion of the Gasdermin protein, forming a pore and killing the cell before phage is ready in the cell. So the same kind of immune system that generates cell death prematurely. So if we're looking at the cell autonomous innate immune system in humans, we now understand that there are many, many components that are shared between our own immune system, the own innate immune system that we have in our cells, and the immune system of bacteria. We realize that the viperin is shared, the SIGA-STING pathway is shared, that the Gasdermins are shared by humans and bacteria. Also inflammasomes, important components, were discovered to be common in bacteria. Tear domains, part of our toll-like receptors, are found in bacteria as well. The RNAi pathway was discovered in bacteria as well, with many different fascinating variations, and many other pathways for defending against viruses. So this picture made us realize that the bacterial immune system has been present in bacteria long before the human immune system came to be. In fact, the bacteria and phages have coexisted, according to most accepted theories, very early on since the initial creation of life. And in bacteria, there has been many immune systems that evolved throughout that time. CRISPR-Cas, restriction enzymes, viperins, the tear domains, Gasdermin, the SIGA-STING pathway, argonauts, the RNAi pathway. At some point, more recently in evolution, but still very far away, two bacteria, or one bacterium and one archaeon, have fused to form the first eukaryotic cell. That's called the endosymbiosis event. And so what we think happened is that the original immune system of first eukaryote was basically comprised of the immune systems that these two cells brought in, so by their fusion. So that was the basis of the primitive cell autonomous innate immune system. And on top of that, evolution generated what we are currently knowing as our own immune system now. So this is kind of a revelation that we very much didn't expect when we started our studies. With this knowledge, we can now try to go back and see if we can generate new knowledge on the human immune system by studying the bacterial immune system. Because so far, what I showed you is that there were immune systems that we knew in eukaryotes. We knew how they work in eukaryotes. We found homologs in prokaryotes, and we verified that these homologs work in the same way. But now, we are trying to start understanding eukaryotic immunity by studying bacterial defense. And very recently, Dr. Francois Rousset from my lab, who's a postdoc, and he is going to start his own lab very soon. He discovered something very interesting in bacterial cells. He discovered a protein domain that is associated with many defense systems, that when the defense system identifies phage infection, this protein domain cleaves ATP from this and removes it from the cell. And as you know, ATP is the main currency of the cell for energy. So, and viruses need ATP in order to replicate. They need it in the cell. So when ATP is eliminated from the cell, the virus cannot replicate. You can imagine it as the battery that the defense mechanism just empties the battery immediately, so the phage cannot use the energy of the cell. And so that was an interesting discovery, but more interestingly, Francois discovered that this protein domain is also found in immune genes of multiple eukaryotes, found in multiple different animals like insects and sponges. And he showed that the same principle of immunity works with the immune proteins of eukaryotes. So here what we found is a discovery in bacteria of immunity that then translated into understanding of eukaryotic immunity. Here's another example of a discovery by Dr. Jens Mohr, postdoc in my lab, that's starting his own lab in Würzburg, Germany, very soon. He studied a 4-gene defense system in bacteria that encodes a protein very similar to an interferon-stimulated gene. And this is a pathway that has the enzymes that are necessary to conjugate ubiquitin on targets. So this interferon-stimulated gene 15 is a ubiquitin-like protein, and if you take the bacterial form, you see that it has 2 ubiquitin-like domains, and you can overlay the ubiquitin-like domains from human ISG15, the human version, it's quite similar structurally. And so in humans, this protein, this ISG15, is activated during infection of viruses and of some bacteria as well. This ISG15 is loaded through an E1 and E2 and E3 enzymes onto substrates to prevent the viral replication. And it is still unknown exactly how this system works in humans. What Jens discovered in bacteria, that this system takes the ubiquitin-like protein, the ISG15-like protein, and conjugates it on the very tip of the tail of the phage. And this generates phages that cannot infect the cell. So here are beautiful images from electron microscopy where Jens tagged this protein by a tag and then sent a gold label antibody to visualize exactly where these proteins are conjugated on the phage. And you see that they're conjugated at the tip of the tail, and this prevents the phage most probably from recognizing the receptor of the cell and infecting the cell. Now, not all the defense systems that we find are homologous to human defense systems. In some cases, we discover defense systems that are fascinating by themselves, but don't look like anything that we know. Here's a beautiful example that we studied recently. It's a defense system that comprises 2 very long genes. One of them contains adenosine deaminase domain, and another contains an ATPase domain. And Philip Kranzush and Brianna Duncan-Lowy from his lab, together with Nitzan Tal from my lab, discovered that these 2 proteins form a huge complex in the cell. And the complex comprises 12 subunits of these that form a core dodecamer. And onto these 12 subunits, there are, each of these proteins produces a 7-mer and heptamer, and each heptamer is sitting on one face of this 12 subunit core, and this forms a 96 subunit complex, a huge complex, which is 4 times larger than the ribosome that produces the defense response in a way that we don't completely understand yet. A kind of fascinating giant complex of immunity in the cell. Here's another example for a fascinating mechanism that's called retrons. And these are defense systems that were studied in my lab by Ati Milman, Maud Bernheim, and Avigail Stoker-Avichai, another super talented PhD student who is starting her postdoc now in NASA's TPAS lab in Germany. And they discovered that retrons, it was known that retrons comprise small RNA that gets reverse transcribed by a dedicated reverse transcriptase to form a hybrid DNA RNA molecule. And what we found is that this defense system guards another defense system in the cell. And when the phage inhibits the first defense system, this retron defense system becomes active and then kills the cell before the phage completed the replication cycle. So this is basically a second line of defense that protects the first line of defense. It senses when the phage inhibits the first line of defense and then comes into action to provide the second line of defense. So I think I will skip a few of these, or maybe just show you one of these. One of the open questions in the field is how are phages being sensed by the defense systems? And for this, Abigail made a huge effort in isolating phage mutants that can overcome defense systems. And she studied multiple different defense systems, and she found that some defense systems sense core replication machinery of the phage, like the DNA polymerase or the single-strand DNA binding protein in order to become activated. Other defense systems sense structural components of the phages, and when these structural components are produced within the cell, the defense system come into action. And yet other kinds of defense systems sense that the phage is taking over the cell, for example, by inhibiting transcription or by inhibiting other defense systems that are present in the cell. But let me skip this part because I think we're reaching the end. So about a year ago, the journal Cell asked us to kind of put all what we know on defense system of bacteria on one poster. This was a nice challenge for Nitzan Tal in my lab. And by now, a year after, this is a pretty much obsolete image because we know of so many new defense systems that are not presented here. But still, it gives you a pretty good picture. And the CRISPR-Cas and restriction enzymes are depicted here, but it gives you a good picture that much of what we know about the immune system of bacteria has been discovered very recently. And so beyond this, even beyond this, there is a set of bacterial defenses. In fact, the vast majority of defense systems that have been discovered, we still don't know how they work. It's a huge, huge, huge list. And this continues, below this slide, a huge list of defense systems that we know they protect bacteria against phages, and we don't know yet how. And this will continue to be interesting for many years forward for myself and for many microbiologists in the world. So to summarize what I showed you that there is an unexpected enormous diversity of microbial defense systems out there. It seems very unexpectedly that key components of our own immune system evolved from bacteria, and we can now start to understand components of our own immunity by studying bacterial defense. So that's the main take-home messages I hope that you take from my lecture. Here's my group of very, very nice people in my lab. So you see that they infect each other by plastic phages. Some of them you see behind me. And I want again to thank you very, very much for listening, and also thank again the HFSPO for awarding me with this super prestigious award. I'm very thankful and humbled. Thank you very much.
Thank you very much, Rotem, for taking us on this fascinating journey of many discoveries and many intellectual challenges and thoughts. Now we have some time for questions and for discussion. So we have an arrangement. Guntram is in the room with the microphone, and I will be watching the audience. And if you want to ask a question, please raise your hand and keep it up until we see you. So I see the first one over there, Guntram. Please introduce briefly yourself. And Rotem, just check, can you hear us well?
I can hear you well.
You
Yes.
need to speak clearly into the microphone so Rotem can hear you, okay?
Can you hear me? Is it good?
Yes.
Okay. I'm Srinivas Alanki. I'm a postdoc at the Francis Crick Institute. I'm a recent awardee for this year. First of all, I'd like to congratulate on your award as well. And it's a fascinating talk and so many new systems to look forward to. My question is about, you've said that any bacterium infected by a phage commits suicide to protect its neighbors of the colony. Do these dying bacteria secrete any molecules to communicate to the neighboring bacterial cells to protect themselves, or just to inform them that there's a new virus on the block?
This is a fascinating question, a beautiful question that we in fact try to see whether bacteria secrete molecules when they die. We so far didn't see this. So we so far didn't see evidence for bacteria alerting the nearby bacteria, but it doesn't mean that this doesn't exist. So we are working with two main lab model organisms, E. coli and Bacillus subtilis, and these organisms, most probably the lab organisms at least, do not alert the nearby bacteria. But I'm pretty sure that in nature, at some point, this evolved, because this would be very efficient way to do a group protection. And if I had to bet, I would bet that it will be discovered in the future that some bacteria do this. So excellent hypothesis.
Hi, Rotem. I'm Beverly Glover. I'm the chair of the Council of Scientists for HFSP. It was our pleasure to decide on the Nakasone Award, and I'm really glad we made the decision we did. That was a fantastic talk. Thank you. All of your work rests on that initial discovery that the genes encoding these different systems are in the clusters in the bacterial genome. And I wondered if you could say anything about, is there an advantage to that in the bacteria? And if so, why isn't it maintained in eukaryotes? Or is it, and we just haven't realized yet?
So again, an excellent question. And to answer this, I will actually share my screen again and try to show what data we have. So one of the open questions that I kind of skipped on is, why do these defense systems cluster next to each other? So I showed you that we discovered this CBAS defense system by finding it next to CRISPR restriction enzymes. We discovered viperin by finding it next to CRISPR restriction enzymes, other defense systems. But what are these defense islands? And this was studied in my lab by Dina Hochhauser, again, a very talented PhD student in my lab. And she discovered that defense systems, the reason that defense systems cluster next to each other is that they are carried by mobile genetic elements, and that integrate these defense systems is in bacterial genome. So if you look at the genome of E. coli K12, this is the lab genome that we use. You see a region of its genome that is lacking a defense system, but you see that different strains of E. coli, other than the lab strain, encode different versions of defense systems, in many cases, multiple defense systems on the same island, that are integrated in the genome by an integrase. And that's something we see again and again. So some conjugative elements carry multiple defense systems on the same island and bring them to bacterial genomes. And so the benefit of it, in order to understand the benefit of it, I need to explain that defense systems come also with a cost. Basically, any immune system comes with a cost. We know that our own immune system gives some kind of autoimmunity. And we also know that, for example, CRISPR-Cas has some costs. CRISPR, we showed that in 2010, a very long time ago, we showed that CRISPR can very rarely, but still can, take pieces of the bacterial genome as spacers and instead of phage genomes. And these pieces are now, the CRISPR system now targets the genome of its own host. And then the host is either killed or needs to pseudogenize, need to delete the CRISPR system from its own genome. And so if you take, and other like restriction enzymes also have some form of toxicity. And it means that if you take a bacterium and put it in an environment where it's not exposed to phages for a while, it will start losing its defense systems. So it will start pseudo-neutralizing its defense system, because competition-wise, if it competes with bacteria that have defense system, it will have more, it will have less cost and will grow faster. But then if the bacterium is moved again to an environment where it's exposed to phages frequently, there is a benefit for opening its gates and acquire mobile genetic elements that bring in new defense systems to the cell. And the fact that the defense systems are aggregating one next to another on the same mobile element is very similar to what we see in antibiotic-resistant genes. So you see that in many cases, antibiotic-resistant genes cluster on a plasmid, on a single plasmid. And so I think the same evolutionary principles contribute to these two things.
I think there was a question over there, right?
I'm Pushkar Sharma from National Institute of Immunology, New Delhi. Fascinating talk. Since I work on malaria parasite, therefore, a very selfish question. Have you discovered some of these systems which may not work as exactly as defense system in unicellular eukaryotes such as yeast, or even to be specific, malaria parasite or other epicomplexan parasites?
Well, it's a great question. And to be honest, we didn't look at the malaria parasite genome. My assumption is that the malaria parasite genome, malaria parasite may be exposed to viruses that infect it as well. And so if you look for homologs of the genes we found, you might find them in the genome, but we haven't looked. It is a very good question.
The mic was being snatched away from me, but the common belief is, as far as my knowledge goes, there's no virus which has been found to be actively infect malaria parasites. So, but your talk clearly sets the stage for another HFSP grant for one of the bright fellows here.
Well, I would be glad to participate in such a grant. I should say that
we got two questions in this corner, and then we go there. So let's.
Sure. Hi, Rotem. This is Paul Turner from Yale University, member of the Council of Scientists, and congratulations on your award. You mentioned a bit about the amazing biodiversity and bioabundance of phages. I wanted to hear a little bit more from you on the need for the redundancy on the bacteria side. What do you think of that, either at a biosphere level, or even if you want to think about human microbiome? And we all know phages are there, but they're very poorly studied. So I want to hear a bit more from you on that. Thanks.
Yeah, so one question that we can ask ourselves based on your question is why do bacteria need so many immune systems? And we now know that on average, a bacterium contains about 6 defense systems. An average bacterium, some bacteria contain as much as 57 different kinds of defense systems that are encoded in their genomes. The reason why bacteria need so many different kinds and so much diversity of immunity is because phages can learn how to overcome some lines of defense. And you can ask the same question, I guess, about cells in our body. Why do we need so many sensors for phage infection and so many effectors to curb virus infection? The reason is that viruses learn how to overcome some of the defense systems. And so if a virus is overcoming one defense system, but you have another one that protects against that virus, it provides a benefit to the cell. And yet still, there are some viruses that can overcome all the defense systems of a given cell and be effective to that cell. In the context of a microbiome, there is a benefit for bacteria having a diversity of immune systems. Also, if you think about the community of bacteria, given that bacteria can acquire defense systems by horizontal gene transfer, so mobile genetic elements could jump between one genome to another, then you can imagine the immune system of bacteria as a pan-immune system, meaning that a bacterium is not only having its own immune system, but it also has the potential to acquire immune system from nearby bacteria in case a phage challenge occurs. And so we introduced this concept of pan-immunity a number of years ago, and I think that data, accumulating data actually supports very much this hypothesis.
Okay. Good morning. My name is Lucy Ochola from Kenya. Thank you very much for your stimulating presentation and for the award. Congratulations. I just wanted to hear your thoughts around the use of bacteriophage. There's a challenge with AMR, and obviously people are advocating, well, some people are advocating for the use of bacteriophage, but based on your own experience and the work you presented, what are your thoughts? Thank you.
Right, so we know that, people have tried to use phages for many, many years to fight bacteria, especially bacteria that are antibiotic resistant. I think that with the knowledge on bacterial immunity, we can now do it in a much more informed way. So I guess one reason why in some studies, it was tested, so phages were tested in the lab and successfully infected bacteria, but in the patient, it didn't work so well, is because the bacteria in the patient may have had different sets of immune systems. So if you have knowledge on the immunity of the bacteria, you can devise a phage cocktail that attacks the bacteria and can overcome the complement of immune system that the bacteria has. And there has been a very promising report from just a few days ago from a company called Biomics that I was the scientific founder of it. Biomics did a clinical trial of a two-part phase 1b/2a trial with CF patients, so phages targeting Pseudomonas aeruginosa in cystic fibrosis patients. And we see definitely that in some patients, the phages in the treatment managed to eliminate the bacteria from their lungs. And we're talking about some people that suffered from bacterial infection for over 10 years and couldn't get rid of it using antibiotics. So there is definitely a promise that will have to be realized by more advanced clinical trials.
Hi. Hello. Okay. Marco Fumasoni from the Instituto Gulbenkian de Ciencia in Portugal. So I think you mentioned that for many of these systems, you can trace back the homology to the bacteria, which is absolutely fascinating. I wonder if you think there are also cases among the various systems of convergent evolution, meaning that, so to say, like independent invention of these systems due to very likely like enormous evolutionary pressure. And linked to this, like, have you considered expanding this data mining to protists and unicellular eukaryotes to see if and how many of these systems could have arisen after the last eukaryotic common ancestor?
So again, both very interesting questions. So for your first question, convergent evolution, I guess you can imagine that it occurs in the level of the concept. So evolution of cell suicide in response to infection, I'm sure that it's evolved in a convergent way. And the same principle of protecting by using signaling molecules seem to have evolved, several times in parallel. In some cases, we can be pretty sure that evolution was actually inherited, so it's vertical evolution, because we can trace the evolutionary pathway. We can see the sequence homology between the proteins or the structural homology between the proteins, and for example, in the SIGA-STING pathway, we see the same enzyme, the same SIGAs, with putting it on the structure of the human SIGAs, we see very tight homology. And these enzymes produce the same signaling molecule and they bind to the same STING domain. And this has unlikely to be evolved by parallel evolution. So I think that in some cases there will be convergent evolution. In some cases, we can clearly say that there has been a direct vertical evolution. As to your other question, there are signatures, and I've seen some papers on fungal genomes that they also contain defense systems like inflammasome-like genes and other Gasdermin genes. I am looking to see how this field will develop. I'm kind of very curious to see how this develops. At this stage in my lab, since there are so many bacterial defense systems that we still haven't explored, and there are so many phage counter defense mechanisms that are waiting to be discovered, we are remaining focused on the bacterial ones, but I hope that our studies will inspire people working on protists and fungi to look for such defense systems in their genomes as well.
Hi, I'm Michal Eisenberg-Bord. I'm a postdoc in Cambridge University. Hi, Rotem. Thank you very much. I have a question. You mentioned the immune systems getting out of control, kind of what we also have autoimmunity, autoimmune diseases. And I was wondering if you can also find like suppressors or inhibitors, and are they also linked spatially in the same areas? And also, can you maybe get more understanding from the bacterial inhibitors on like what's happening in eukaryotes and humans?
Right. So in fact, yes. Because immune systems are dangerous to the cell if they are going out of control, what we find is that they are expressed to pretty low levels, but they're still expressed in the cell before the phage infects. And the reason is that phages are pretty rapid. So some phages like T4, once it infects the cell, after 4 minutes in 37 degrees, it degrades all the DNA of the cell, of the bacterium. So this leaves too little time for the bacteria to sense the phage and then activate expression and then produce the protein. So the proteins must be there before the phage infects, and the sensing must be done in the level of the protein. That said, we do see, or it's studied by others, not myself, we do see that there are inhibitors, that there are repressors for expression that allow more expression, so more dramatic expression of the defense genes when signature of infection occur. For example, when DNA breaks are sensed within the cell, when the cell senses that its DNA is becoming broken, it activates the expression of some of its defense systems. And the reason is that DNA break is a very strong signal for prophages, for dormant phages to become active. So that's what we know about inhibitors of infection. In other cases, there are a set of proteins called toxin-antitoxin systems, where the toxin is supposed to kill the cell, but the antitoxin kind of holds it together. And when a phage infects, there is something that lifts the antitoxin. The antitoxin recognizes the phage, lifts away, and then the toxin is doing something toxic to the cell and kills it. So you can imagine this as an inhibitor. I don't know of a version of that thing within the human immune system, but I imagine that the same principle may be present in human immunity as well.
Rotem, I don't see any more questions from the audience. There is one more just now, so please.
Thank you. Hi, Rotem. My name is Katie Gazetta. I'm a postdoctoral researcher with Randy Platt at ETH Zurich. My background is really in the gut microbiome, and I know this is very related to phages. And specifically, my question's about the geographical biodiversity of phages across gut microbiomes across the world. So my question relates to one of our more recent studies that is coming out of some of my PhD work on how when you stress mice psychologically, their phagome changes in the gut microbiome. There's a change in the phages that are present. And when you actually take phages out of their gut microbiome before you're stressing them and you give these phages back to them, you're able to ameliorate some of this stress phenotype just through this phage transplant. So based on this, and this was done in lab mice, I'm wondering if there's much known about the defense mechanisms of bacteria across the globe. So whether phage treatments would work, do you think that they would work in populations in the US the same way that they would work in populations in Asia or Africa, or if you'd have to kind of isolate within communities? Do you think, is there much known about this yet? Thank you.
Yeah, so I mean, the study that you described sounds really fascinating. I'd be very glad to know, kind of the molecular mechanisms guiding this. I can say that there are tools these days that if you have a genome of your bacterium, you can you can send it to the online tool, and the online tool will tell you which defense systems that are known are present in that genome. And we see that defense systems are among the most rapidly diversifying elements in bacterial genomes, meaning that the different strains of the same species will have different sets of defense systems, even if the strains live, together in the same environment. So the diversity of this pan-immune system is something that we see very strongly at the strain level. This is different when we're talking about pathogens that are clonal and spreading in the world. For example, Vibrio cholera, it's a clonal pathogen, and it has a specific, or at least the seventh pandemic clone has a specific set of immune systems that many of them have been deciphered. And so for this specific clone, I think that you can devise a specific set of phages that will most likely work in different places in the world. For communities that are spread globally, I imagine that you'd want to sample the community in different parts of the world and see whether they have different sets of defense systems. And if there are, you probably want to diversify the mode by which you study their phages.
Thank you.
Thank you, Rotem, again. To close the discussion, I would like to ask the final question, which is more about the scientific journey rather than the specifics. Until short, say 10 years ago, the common wisdom and knowledge accepted was that the immune system of bacteria can be explained by CRISPR-Cas. That was the starting point. So what was your aha moment? What was your spark, which makes you to distrust it? I mean, why did you start to look around it? Because that was a common wisdom. So what was that spark which brought you into this new avenue?
Well, I think it. So first of all, we studied CRISPR-Cas since very early on. So I think our first paper was 2007, and we were interested in this fascinating immune mechanism way before it became interesting because of its biotechnological promise. And first of all, it always stood out to me that CRISPR, although it's very efficient in defense against phages, most bacteria don't have it. And so that was kind of, a red flag, for myself, because we know that most bacteria are infected by phages. So how come there are bacteria, or even most bacteria, that don't have CRISPR? And so that was an initial thing that kind of sparked our interest. And the second one is that the late discovery of CRISPR was another thing that, was a bit awkward, given that for so many years, phages have been the working course of molecular biology in the world. And so realizing that CRISPR is not present in the model organisms that we use, or it's not active there, and that in fact, the fact that we use bacteria as model organisms in our lab is because they accept DNA very easily, meaning that they must have fewer immune systems than normal bacteria in the world have, kind of led us to think that there might be immune systems that are found in non-model organisms. So people, including ourselves, we really like model organisms, because in model organisms, you know what each gene does, or many genes do. You can rely on studies of others. It's very convenient. But we found that in order to really discover fundamentally novel genetic systems, we had to go out of our comfort zone and kind of wander around the unknown part of the non-model organism genome. And this, very luckily brought very interesting discoveries for us. I should confess that there are many multiple studies that we did in our lab that started with hypotheses that were similarly exciting, but then we couldn't verify this hypothesis. So we just wasted time, running after something that we couldn't verify. But I think that's what's fun about science. I mean, if you allow yourself to really go into the darkness in terms of knowledge, the darker part where knowledge is not there, and and put a small flashlight and start shedding light on areas of darkness and knowledge, that's when you have a chance to find something fundamental that was not known before you. And that's, I think, as scientists, we want to strive to go into these places.
Thank you very, very much again. We do hope that safety and peace will return soon to your region and to your county, to all the people, and we hope to see you very soon again amongst us. Please join me again thanking to Rotem for this fascinating journey through the stars.
Thank you very much.